Hello and welcome, everyone. For those of you who don't know me, I'm Paula Hammond, and I'm the department head in chemical engineering, which is the home department of our honoree, Bob Langer. It is my honor to be here today to celebrate Bob Langer on his receipt of the Dreyfus Prize for his extraordinary achievements in the advancement of human health. The Dreyfus Prize is perhaps the most prestigious and important award for achievements in the chemical sciences, recognizing transcendent scholarship that illuminates new ways for the work to improve human lives. To say that Bob has touched many lives is an understatement. It is difficult for our students today to understand that when Bob began his career in the 1970s, chemical engineers were not routinely viewed as key participants in the biomedical sciences. Bob had to create that mold for himself, overcoming early obstacles and proving his value as a collaborator to clinicians and the value of the intersection between engineered materials and the medical field as a whole. The end results speak for themselves. Angiogenesis inhibitors for tumor treatment have been delivered to over 20 million people. That's Bob Langer innovation. Pioneering work on microspheres and nanoparticles for controlled release drug treatments for schizophrenia and diabetes. That's Langer innovation. Bioerodible polymer wafers to deliver localized chemotherapy for brain tumors. That's Langer innovation. 3D polymer scaffolds to grow new skin cells for burn victims. And these are just a few early examples of Bob's research that are already touching millions of lives. And in fact, we're just seeing the tip of the iceberg. Now he is developing tortoise-like oral capsules that flip into place to deliver micro-injected insulin straight to the gut. He's using machine learning methods to devise better drug formulations. Technologies are emerging from the Langer lab today that will become the standard of care in treating diseases for decades to come. And I could go on, but I don't want to steal Bob's speech. Bob's impact as a researcher is only surpassed by his impact as a mentor and a teacher. Almost 1,000 students have passed through the Langer lab since he joined MIT as a faculty member in 1977. Bob has always been known as someone who encourages his students to follow their passions, to fearlessly tackle seemingly unsolvable problems, and to take their discoveries out into the world for the betterment of mankind. He has an incredible life in perspective. He is never afraid to ask why not, and he teaches that to his mentees. Bob's former students and postdocs are leaders in the academy, in industry, and in the startup world. The Langer Lab's family tree is vast and growing and will touch human life in ways we can't even imagine today. In short, Bob is worth celebrating, and I want to thank the Dreyfus Foundation for recognizing him with this extraordinary honor. The Dreyfus Foundation holds a special and revered place at MIT and in the broader chemical sciences community, and so many of us have been the beneficiaries of their support of our field. I want to thank President Hank Walter, Scientific Advisor Matt Tirrell, Executive Director Mark Cardillo, and all of the members of the Dreyfus organization who are here with us today. I'd like to close by speaking to the students here today who are just beginning their careers and discerning where their life's work will be. I hope that when you see Bob, you will begin to imagine the possibilities that are open to you through the study of the chemical sciences and engineering. They are truly limitless, and they provide unparalleled opportunities to improve human life. It is my great good fortune to work with colleagues who show me that reality every day, colleagues like Bob Langer. Thank you, Bob. With that, it's my honor to welcome to the stage the president of MIT, Rafael Reif.
Thank you, Paula. For those of you who may not know Paula well, I would just like to say that in addition to being an exceptionally accomplished scientist and inventor, she's also an incredible citizen of MIT. She has also been here for a while. Like Bob, Paula earned her PhD from MIT. And like Bob, she also had the good judgment to attend MIT as an undergraduate. But since Bob has blessed our community with his presence for quite a few decades, I believe we can forgive him for his 4-year flirtation with Cornell. Let me begin with a warm welcome to the representatives of the Dreyfus Foundation, President Henry Walter, Chair of the Scientific Affairs Committee, Matthew Tirrell, and members of the board of directors and scientific advisors. You're here to confer upon Bob Langer a remarkable distinction. The Dreyfus Prize honors Bob's exceptional accomplishments, a career of countless breakthroughs, using chemistry to advance human health. This evening, I know we will get a vivid sense of Bob's immense impact on the world. But as we begin, I would also like to reflect on Bob's impact at MIT. As many of you know, after receiving his PhD in chemical engineering, Bob had a great many job offers from the oil industry. Fortunately for all of us, he turned down every one. Even back then, Bob wanted to use his training and expertise to have a direct impact on improving people's lives. So after meeting leading cancer researcher Judah Folkman, Bob began pursuing his own pioneering research at the intersection of chemical engineering and medicine. Now at MIT, we like numbers. After all, we are in 26,100. So here is a quick numerical expression of Bob's career. More than 1,400 articles, more than 1,300 patents, more than 220 major awards, and as Paula said, 1,000 students who have begun their careers in his lab. Think about that last number, almost 1,000 students. Bob's accomplishments as a scientist, engineer, and inventor obviously put him in a category all his own. But of all of the gifts Bob has given the world, the most beautiful may be that he has trained and inspired so many brilliant young people. These Langer Lab graduates are leaders, not only in medicine and academia, but also industry and government. They are working to prolong life and relieve human suffering around the globe. And this means that even when Bob is sleeping, which honestly cannot be very often, the global alumni of the Langer Lab are hard at work somewhere in the world. Most academics, even very ambitious ones, would be very proud to say that they had mentored a new generation of scientists and advanced research in vitally important fields. But Bob has gone way beyond that because he helped create new fields too. And that is why we're honoring him tonight, for developing novel drug delivery systems that have revolutionized how drugs are administered, and for pioneering the field of tissue engineering to address medical conditions that cannot be treated with drugs. By some estimates, the innovations and discoveries Bob has shared with the world have benefited as many as 2 billion people. I expect Bob is not completely satisfied with that numerical accomplishment either. So we can only wait to see what he comes up next with. At MIT, with our unusual concentration of unusual talent, Bob stands out as not just unusual, but spectacular. Bold, down to earth, and incredibly creative. A daring inventor, a brilliant entrepreneur, a beloved educator. Bob Langer teaches us all through his example, and he truly represents the very, very best of MIT. So it's deeply fitting that the Dreyfus Foundation is recognizing him tonight. The foundation has contributed so much to the Institute. 2 named professorships in the chemical sciences, wonderful support for young faculty through the Teacher Scholars Awards, and of course, the Camille Dreyfus Chemistry Building, a hub of innovative research and discovery and the headquarters of chemistry at MIT. Over its 70-year history, the foundation has had a profound impact on MIT, and we are profoundly grateful. I would like to turn the proceedings over to the president of the foundation, Henry Walter, who will present the Dreyfus Prize. Henry?
Thank you, Dr. Reif, for your kind introduction, and thanks to Dr. Hammond also. We are grateful to all at MIT who have helped produce this event to honor Dr. Robert Langer's remarkable achievements with the 2019 Dreyfus Prize. The Dreyfus Prize is awarded this year for chemistry in support of human health, and it is a pleasure to make this award to Bob Langer, a true giant in this field. We are delighted to be at MIT, where the Dreyfus Foundation has a long history. Our institutions have many notable connections, such as the Dreyfus Building and the Camille and Henry Dreyfus Endowed Professorships that are currently held by Dr. Stephen Buchwald and Dr. Christopher Cummings. Starting in 1955, MIT faculty have received over 100 awards from the Dreyfus Foundation, more than any other institution. This includes 29 who have received the Dreyfus Teacher Scholar Award, which supports outstanding young researchers. In addition, several MIT faculty and alumni have made valuable contributions to the Dreyfus Foundation's mission over the years. When I joined the board in 1978, Professor Robert Alberti was one of our key advisors, and he served over 30 years in that capacity. Today, Dr. Arup Chakraborty and Dr. Joanne Stubby also serve as Dreyfus advisors, and Laura Kiesling serves as a consultant. Given this rich history, it is especially rewarding to be here at MIT to present the Dreyfus Prize to Bob Langer in the company of his family, friends, and colleagues. The prize recognizes an individual for exceptional research in a selected area of the chemical sciences. The previous award winners shown on the screen are chemists who represent a range of diverse and extraordinary achievements. We are pleased that George Whitesides and Christopher Makasevsky are with us today. George and Chris, would you please stand? Yeah. Where'd George go? There he is. Bob Langer's career has been equally distinguished. His innovation have brought enormous benefits to humanity. The vast scope of Dr. Langer's accomplishments epitomize the ideal person the prize was established to recognize. I am sure the Dreyfus brothers would be proud of his astounding productivity and ingenuity. Professor Langer, would you please join me? Jerry Brandenstein, our Associate Director, will read the citation.
The Dreyfus Prize in the Chemical Sciences is awarded to Robert S. Langer for his discoveries and innovation of materials for drug delivery systems and tissue engineering that have had a transformative impact on human health through the chemical sciences.
On behalf of the Board of Directors of the Camille and Henry Dreyfus Foundation, I'd like to present this medal to you as the winner of the 2019 Dreyfus Prize in the Chemical Sciences.
Thank you so much. Thank you so much.
Congratulations. And not to be forgotten, I'm glad somebody laughed. Here is a check for $250,000 representing the monetary part of the award.
Thank you
So
so
again.
much for that, sir. Thank you.
Matt Tirrell will now introduce Professor Langer. Dr. Tirrell is the Dean of the Institute for Molecular Engineering at the University of Chicago and a director of the Dreyfus Foundation. Matt.
There's a great deal of happiness and a certain trepidation being the fourth person to speak about Bob before he speaks. But I'm very happy to have the job of introducing Bob Langer for his award address on accepting the Dreyfus Prize. I'll try to give just a few very succinct points that haven't been made quite so sharply so far. Materials with chemical properties have had a transformative effect on human health. And Bob Langer did the seminal work that started this revolution, period. His discoveries have led us to understand how they control the release of therapeutic molecules through materials and how to create new tissues and organs. Thus, Bob created at least two distinct and gigantic fields at the intersection of chemistry and medicine: controlled release materials for delivery of therapeutic molecules and tissue engineering, and more. His inventions have been translated, often by Bob himself, into many products that profoundly affect human health. Before Bob's research, scientists used off-the-shelf polymers to address a biological or medical problem, even though the polymer was not designed for those purposes. Bob proposed a new approach that provided the first rational framework for biomaterials design. He asked, what are the important properties and qualities desired from the polymer from the viewpoint of chemistry and biology and engineering? He then specifically synthesized polymers for the specific application. In my opinion, which I'm sure is shared by countless others, he is the very top chemical engineer in the world working in the medical sciences, as evidenced by his achievement and impact in science and technology. I have a few statistics that President Reif didn't mention. Bob is the most highly cited engineer in the world, with well over 250 citations to his work and 110,000 just since 2014, and a nearly unfathomable H-index that's fast approaching 300. For those of you not in the know, H-index is something that we academics compare one another with. Something like 300 companies have licensed his inventions, and roughly 40 companies have been spun out of the Langer Lab during his career. And this has been mentioned, more than 300 of Bob's graduate students and postdocs now serve on the faculties of major universities. Overall, an unparalleled record. We've heard that Bob earned his undergraduate degree at Cornell, but since then he is almost continuously a homegrown product of MIT, starting as a graduate student in the early 1970s, and I'm proud and humbled by the fact that I've known and considered Bob a friend for a good fraction of that time, certainly for the last 40 years or so, since the late 70s. One personal memory that stuck with me is that I believe Bob was the first person I knew who had a mobile phone. I remember his using one in the early 1990s, maybe around 1990, when we were both on the External Advisory Committee of Georgia Tech. Bob's phone was a little bit like Gordon Gekko's, if that means anything to you, but he had one. And this early adoption of technology has led to one of Bob's most endearing personal characteristics. If you email or text Bob saying you'd like to speak with him, you will hear back from him within minutes, not hours. And this speediness is just one manifestation of the generosity that accompanies Bob Langer's technical brilliance and unmatched record of accomplishment. Ladies and gentlemen, I now turn the floor over to the person we are honoring today with the Dreyfus Prize in the Chemical Sciences, Robert S. Langer.
Well, thank you all so much. I feel very humbled to see so many friends and family and colleagues here. It's very nice of you all to come. And so I thought what I'd try to do is mostly tell you how I got started and some of the more recent things we've done. So a little bit about my background, some of which you heard, but I got my degree, actually, even though I was an undergrad at Cornell, I did get my doctorate from MIT, which I got in 1974 in chemical engineering. But then, not unlike a few years ago, there was a gas shortage. And what happened is pretty much all my classmates around that time, 1973, 1974, they'd go to oil companies because they had so many job offers. And I thought that, that I should do what my friends did. So I applied to companies, mostly oil companies, and I got 20 job offers, 4 from Exxon alone. And really, I wasn't that good. But they had so many job offers and so many openings. And I still remember one of them. I remember going to Exxon in Baton Rouge, Louisiana. And when I was there, one of the older engineers said to me that if I could just increase the yield of this one petrochemical by 0.1%, they said that would be wonderful. That would be worth billions of dollars. And I remember flying back to Boston that night thinking to myself that I really didn't want to do that. And it made me think more about what I did want to do. And I always tell my students that, it's very useful to know what you don't want to do as well as what you do. And one day I saw an ad in a chemistry journal to be assistant professor of chemistry curriculum development at City College of New York. And I was very excited about that because when I was supposed to be doing my doctorate at MIT, one of the things I spent a lot of time doing was help start a school in Cambridge for poor high school students, the group school. And I got very involved in developing new chemistry curriculum to try to make chemistry more fun and interesting. And I love that. And so I wrote City College of New York a letter applying for that job, but they didn't write me back. But I really liked that idea, so I found all the ads I could to be assistant professor at different colleges to develop new chemistry curriculum. I found about 40. I wrote to all those places, and actually I don't think any of them wrote me back. So that wasn't going so well. But it did make me start to think, I'd really like to use my chemical engineering and chemistry background to help people. So I thought, what other way might I do that? And I thought about medicine. So I started writing to a lot of hospitals and medical schools. And they didn't write me back either. And then one day I remember going into the lab. I was getting pretty discouraged. And one of the postdocs in the lab, Barry Bunow, said to me, he said, Bob, he said, there's this surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. So I wrote to Dr. Folkman applying for a job, and he was nice enough to offer me one. And I'll just show you, he had this idea. Let me just see if this, and his idea is shown here. This is actually from the New York Times in the 1970s. And his idea was that the way a tumor grew was, it's actually sort of a chemical engineering problem. A tumor starts out abnormal, and then it grows as this three-dimensional mass. It won't get larger than a millimeter cubed, except for the fact that he proposed that the tumor sends out this signal, which he called TAF, or tumor angiogenesis factor. And what that does is it causes the blood vessels, the surrounding blood vessels, which are normally stagnant, to all of a sudden start growing right to the tumor. And that solves the nutrition problem for the tumor. And the tumor gets bigger and bigger and ultimately can spread through those blood vessels. That's a process called metastasis, and that can often eventually kill. And his idea was that somehow if you could stop blood vessels, that might be a new way to think about stopping cancer. I thought this was terrific. It turns out that he was widely criticized for this idea, but I thought it was a really good idea, and because I didn't know any biology. I still haven't had a biology course since 10th grade. But at any rate, he said, well, would I come there and try to actually, in so doing, try to see if we could try to prove that this was right, and in so doing, isolate substances for the first time that might stop blood vessels from growing. So how do you think about a problem like this? Well, we kind of broke it up into two parts. First, where would you find such a substance? So Henry Brehm, who was a medical student at the time, now head of neurosurgery at Johns Hopkins, had done some work on cartilage and from baby rabbits. And what I started to think about was, well, we couldn't really get enough cartilage from baby rabbits to try to address, how substances that could get blood vessels from growing. But one of the things that you learn in chemical engineering is scale up. So I went to cows. I found a slaughterhouse in Cambridge, and I got a cow bone, but still one cow you can't do that much with either. So I found out, where do all the cow bones in northeastern United States go? And it turns out, I found out they go to these meat packing places in South Boston. So I made an arrangement with them to get all their bones. And I would drive there 4 times a week, bring them back, and start scraping the meat off the cartilage. And this is actually one of the bones that I've got. And I'm actually very proud of this. It doesn't start this way. There's meat on it, and you use what's called the periosteal elevator to scrape it off. And then it gets like this. Then you cut this off. And then we develop these extraction procedures with guanidinium hydrochloride, and then various purification procedures for different types of column chromatography. And at the end of several years, I had, say, 100 fractions. And then that brings up the second question. How could you test whether any of these different fractions from cartilage worked or not? So what you need is a bioassay. And if one looks back at the history of various biomedical things, one of the things you often see is when people isolate factors, well, probably the most rate-limiting thing is having a bioassay to study it, some way to really examine how blood vessels grew. And at that time, there was no bioassay, so we had to invent one. And the bioassay that Dr. Folkman and I and Henry and others would try to develop is shown here. The idea is that we'd have a certain type of tumor, V2 carcinoma, you could put in the rabbit eye. And then our idea was that we'd develop a slow-release polymer that A, would be inert in the eye, in other words, not cause any harm. That's very difficult. But 2, it had to be able to release molecules of any size, including large molecules, which were in cartilage and other places. And that had never been done before. And in fact, so Dr. Folkman was on the board of the one company in the world that at that time was doing any work at all on delivery of molecules. And that was a company called Alza. And he went there and he asked people if they could help. But they said, well, you really can't do that because large molecules can't slowly diffuse through solid polymers any more than, say, any of us could walk through a wall like that. The literature actually said something very similar, that the use of polymer matrices for slow-release systems has been virtually restricted to small molecules. And really, I think the only thing that Dr. Folkman and I had going for us is that we hadn't read any of this. So I tried anyhow. I spent about 2 years working in the laboratory, really kind of almost Edisonian-like, experimenting with different techniques. And I actually found over 200 different ways to get this to not work. But eventually, we were able to make little microspheres. Here's a picture of one. Here's one cut in half. And now you can adapt these procedures to make nanoparticles. And we published in Nature in 1976 that you could use this approach to release molecules of any size. Now, these early systems, the release rates are not constant, but using some modeling techniques in chemical engineering, we figured out ways that we could get the right drug distribution and geometries so that we could, in fact, get constant release. And without going into that too much, just as a proof of principle, here is an example where we released a protein albumin for about 55 days. So, you know, one of the things that you have to start doing when you're in academia is you give presentations. And so in 1976, I got asked to give a presentation for the first time. It was actually in Midland, Michigan. That's a big polymer center where Dow and Dow Corning was. And so I had to give this 20-minute talk, and I'd never really given a major talk before. And so I practiced this talk for a while. And then the day came, and I had to give this talk, and I was like a lot younger then, and it was to a very distinguished group of polymer chemists and engineers. And I gave the talk, and I thought I did all right. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk, all these older scientists, being nice people, would want to encourage me, this young guy. And when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, "We don't believe anything you just said." And as they pointed out, just like I mentioned, it was in the literature before, that large molecules can't slowly diffuse through solid polymers. And they also pointed out that organic solvents would denature whatever we put in. And actually, things kind of went downhill from there. I really liked being a postdoc at Children's, but one of the things that I wanted to start doing was getting some grants. So I'd write grants to the government, and I got my first 9 rejected. In fact, very soundly. I remember one I wrote, and this was actually on some cancer work, and they wrote back that Dr. Langer shouldn't get this. They said he knows nothing about biology and even less about oncology. And I knew enough about math to know that probably wasn't possible. But but it didn't really matter. I wasn't doing very well. It was like a pathology study section. So at any rate, so that wasn't going so well. And then also, you know, I like I said, I really liked being a postdoc, but a lot of my friends said, you know, you shouldn't do that forever. So I started applying for faculty jobs, and I'm a chemical engineer, so I applied for faculty jobs, and actually not a single chemical engineering department in the country would hire me. They basically thought this kind of bio stuff that I was doing didn't make sense. In fact, in some of the introductions, I appreciated hearing that. You know, now I take some satisfaction that probably half of the chemical engineering departments in the United States actually have bio in their name, but not ours, but a lot of them do. And you know, and so that you know, it's kind of nice, but that wasn't exactly like that in the 1970s. So, but anyhow, so I didn't get any jobs there. But Judah Folkman knew Nevin Scrimshaw. Nevin was head of the Nutrition and Food Science Department at MIT, and he introduced me to him. And for those of you who knew Nevin Scrimshaw, who was also an institute professor, who's a very famous nutritionist, he liked me, and but he was kind of what I call almost a benevolent dictator kind of department head, meaning that he liked me, so he offered me a job, but he didn't bother to ask the rest of the department what they thought. That probably would have been okay except for one thing, which was that the year after I joined that department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. I'll back that up. A couple of years ago, my friend Mike Marletta, who later became head of the chemistry department at Berkeley, a member of the National Academy of Science, I was getting some prize, and he gave a speech about it. And I'll just do a quote from his speech. He was talking about what it was like to be with me at MIT when he was on the faculty there in the nutrition department back in the 1979 and 80. And I'll just read it to you. He said, "One evening, I went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors." He said, "A senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for polymeric drug delivery, he blew a cloud of smoke in my face and said, 'You better start looking for another job.' Mike said he thought he was in a Fellini movie." So that wasn't so good either. But anyhow, I kept trying to work on this, and one of the things that we wanted to better understand was how did this work, since people didn't think it could. And so what I did is you know, one of the things they have in surgery departments at Children's Hospital and MIT is they had a cryomicrotome, so you could cut thin sections, usually through tissues. But we started using it to cut through polymers. Here's a 5-micron thin section of one of the polymers that we were using, ethylene vinyl acetate copolymer. There's no drug in it, and if you did a diffusion experiment, you'd find that molecules just a few hundred molecular weight, like 200 molecular weight, would not get from one side to the other. But it turned out that when we put a red protein, just to visualize it better, this is myoglobin, and now we cut a thin section before any release, you see what we call a phase separation. The polymer is whitish and the drug is reddish. And if you release this for, say, a year or more, it looks very different than what you saw 2 slides ago. Left behind where the drug was are these pores. And these pores, it turns out, are large enough so that molecules, even millions of molecular weight, can get through. But they also are interconnecting, and there are tight constrictions between them, and they're also incredibly winding and tortuous, so it takes a really long time for the molecules to get through. One way I sometimes explain this to people when I give lectures around the world is I say it's kind of like driving a car through Boston. There's a term we use in chemical engineering called tortuosity. And Boston and these systems have very high tortuosity. And over the years, a number of our students, like Larry Brown and Mark Saltzman and Ron Siegel and others, did a lot of mathematical modeling so you could predict how to control the tortuosity. And so by doing it, you can really design these to get almost any release rate you want. And since you could do that, you could go back and now try to solve the angiogenesis problem. So this was the assay that we were developing. And this we published in Science. And I probably should give a warning. I'm going to show you a picture. We actually did about 2,000 rabbit eyes, 1,000 rabbits, and testing many, many fractions, almost all of which did not work. And I kind of think of those as the controls. And they really are, if you think about it. They're actually isolated from the same type of tissue, but just purified differently. And almost every one except one did not work. But, and I apologize, if anybody, this is very graphic, so some people may not want to look. But we did, I'm just going to show you representative eyes from the science paper. And here is, at 9 weeks, if you do a control, this was out the cartilage-derived inhibitor. Here's the tumor. Here's the polymer. You can just see the tip of it. But you see the sheet of blood vessels growing over it. If you looked at this eye 3 weeks later, it would be three-dimensional, out of the orbit of the eye. It would actually kill. We sacrificed them before that. But if you put the inhibitor in, it's very strikingly different. The vessels are lower, they're sparser, and they actually avoid the polymer. So, and actually, 100% of the time, these take off and become three-dimensional. About 60% of the time, these never did, which was striking. So that actually showed for the first time that you could now have these diffusible inhibitors and even isolate fractions. But it still took another 28 years from this paper before the first inhibitor would be approved by FDA. And that only came about really, I mean, people use the techniques that we developed, like these assays, but it really took the work of people like Genentech and other companies, and ultimately billions of dollars to isolate the first inhibitors. But starting in 2004, something remarkable happened, 28 years after this. And this may be hard to see in the back, but what I wanted to show you is starting in 2004, one after the next angiogenesis inhibitor is approved by FDA. Avastin, which was the first of these, that was from Genentech, is probably the #1 or 2 best-selling biotech drug in history. But there are many, many others that have been approved for different kinds of cancer. And actually, the major ways of treating eye diseases like macular degeneration and diabetic retinopathy, for which before this there were no pharmacologic treatments, now there are actually ones that can even in some cases reverse that, like Lucentis and Eylea. So that was exciting to see eventually that something might happen. I mean, medicine, unfortunately, moves slowly, as anybody working in that field knows. Nonetheless, it was wonderful to see that these are around now. We also thought that maybe we could use these technologies not only to study different informational macromolecules, but also to deliver molecules that might have short lifetimes or where patient compliance might be bad. So Larry Brown, one of my graduate students at MIT, did a study where we made an aspirin-sized pill, actually did his whole thesis on this, I'm just going to show one graph, but put it underneath the skin of a diabetic rat and showed that you could lower the blood sugar for over 100 days. And one of the things that Dr. Folkman said to me one day is that we should file for a patent on this. That might seem very common today, but it wasn't then. In fact, Children's Hospital never had a patent before this. So we filed for a patent for the first time, and actually 5 years in a row, 5 times in a row, the patent examiner rejected it. And he kept not really understanding it, in my opinion, but it didn't really matter. He was calling the shots. And the head of the hospital came to see me and said, you know, we should just give up because the guy's never going to allow it. But I don't like to give up. And I started to think, how could we convince the patent examiner to allow this patent, legally, of course? And science really wasn't working. But as I told you all, when we first started doing this, people said this wouldn't work, and it was impossible. I wondered if anybody ever wrote that down. So you can do what's called a science citation search, meaning I could look back at my 1976 Nature paper. I did this in 1982. Look back at it and see who wrote about it. And actually, I did that, and I was very surprised. I found actually a number of good quotes, but this in particular I thought was very helpful. It was by 5 of the top polymer chemists in the world. I'll just read you what they wrote describing the field. They wrote, generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising, so that was a really good word for the patent examiner. I've reported some surprising results that clearly demonstrate the opposite. So I showed that to the heads of the hospital, and they were very excited, flew down to Washington to see the patent examiner and showed him this. And the patent examiner said, gee, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote this. It's a true story. I wrote them, and they were all nice enough to write me back that they really did write it. And then we got this very broad patent, and with that, we would either license it to companies I got involved, as people know, starting companies. We taught different companies how to do it, and today that's led to many, many products, just a few that are shown here. I mean, examples are if you had a luteinizing hormone, releasing hormone analog, which is a peptide, you know, people tried to deliver it orally or nasally, but they could hardly get anything in. And then when you injected it, it was destroyed right away. So they put it in little microspheres, and now it's injected. And originally the company, Takeda, that made it, which we had an arrangement with, they made it last for a month. Now they can make it last for 6 months. And there are many others too. I mean, in fact, some of the major ways of treating opioid addiction, Vivitrol and others, are based on this. Ways of delivering drugs to treat schizophrenia, stop bleeding, different eye diseases are all based on these ideas, and they end up being used by tens, if not hundreds of millions of patients every year. So, so this is kind of how I got started, and a lot of these keep getting approved. Even last year, one was approved for arthritis and so forth. But one of the things that we started to do is we've also started to think, could we ever deliver these more complex molecules orally? Actually, even before I go into that, I probably should divert and say some of the things that this would start to lead us to. So among the different projects that this launched was, you know, one of the things we've been doing a lot recently is the last 5 or 6 years working with the Gates Foundation to develop new medicines, new drug delivery systems for people in the developing world, new kinds of vaccines, new kinds of pills, new ways of giving nutrition. We've also worked with Mike Sema to develop smart delivery systems like microchips that you could turn drug delivery on and off. And with Dan Anderson and Omid Farrokhzad to develop new nanoparticles that might be able to deliver siRNA, messenger RNA, anti-cancer drugs, and other things. But one of the things I thought I'd highlight a little bit today, because it follows some of what I just went over, is could you ever deliver these more complex molecules orally? And here, Gio Traverso, who's actually a gastroenterologist and molecular biologist, but the mechanical engineering department actually was, and you'll see where some of the mechanical engineering comes in, was smart enough to hire him as an assistant professor, joined our lab, and along with Alex Abramson, who is a graduate student, Esther Calvado, who's a postdoc, Nicholas Rockstead, who is a visiting scientist from KTH, we tried to come up with a system that might even be able, you could swallow, that might be able to even deliver complex molecules orally. Now that might seem next to impossible. How could you deliver a large molecule like insulin orally? And we wrote an initial paper with Dan Blankstein, Dan Anderson, and others, with some microneedles showing that we might be able to do something. And then Novo Nordisk, a company, read that, and they called up and said, well, could they get involved? And they gave us a big grant. So the idea, though, of course, is that if you try to deliver a large molecule orally, it's very hard to get through the intestine, and then they also get degraded by enzymes. Now, different groups, including our own, had tried to do this before. You might be able to put some type of transporter molecule on the drug. And one of the things that's very exciting, in fact, it just got approved, I think Monday, is what Novo Nordisk did themselves with what's called semaglutide. That just got approved by the FDA, and that's a peptide. But what's amazing is if you look at its bioavailability, it's, if you compare injection to orally, you have to give 280 times more orally than what you can get by injection. In other words, you probably get about way less than 1% in. That being said, even doing that badly, that's going to be a multi-billion-dollar drug. It's really going to change diabetic treatment. But we'd like to do a lot better. How could you do that? So one of the things that I mentioned that Gio Traverso, he's a gastroenterologist, and people, as a gastroenterologist, one of the things he's pointed out is that people actually inject things into the gut now. Of course, they go through your back end, not your mouth. But when you do that, it's very high delivery efficiency. It's not really painful, and you get rapid tissue regeneration. So the question is could we swallow something and then have it inject something into the stomach? So we've designed this pill to do this, and I actually brought one. It's kind of tiny. You could actually put it in a capsule. It's this big. There's actually a little hole in it. And one of the things that Gio and Esther and Alex and Nicholas did is they've been able to make needles. But the needles they make could be 100% insulin pretty much, or very close to it. Now, the needle, you of course don't want to just shoot the needle out right away. But what's behind the needle is a spring, right? So you could cock the spring, and the idea is as soon as you have the spring cocked, and it's going to shoot the needle out into whatever tissue it is. But of course, you don't want that to happen right away if you're swallowing it, right? Because it's going to do it in your mouth. So what we did is we put it in a sugar glass. So there's a sugar glass inside this where the needle and spring are embedded, and that fixes it. So nothing will happen, but the point is that by osmosis, and you can control this, I'll go over by how much you put in about 30 minutes, and we could make it 40 if you wanted, but about 30 minutes, that sugar glass will break. And when that breaks, the spring is released, it shoots the needle right in. Now, but you might be thinking, well, okay, that's going to be in the stomach then, but how do I know that the right side is going to be facing, how do you know this is going to be facing the right way? So one of the things that the team did is they designed this little system. Again, this is some mechanical engineering, like what's called a leopard tortoise. So no matter what you do, and this is going to probably be hard to see, but I could have a volunteer, but what you could see is no matter what you do, this will always land on the face because of the weight distribution. So it will always do this no matter what. So therefore, it's going to be facing the stomach. So just to show you then what happens, this kind of self-orients, and then it shoots out just like the needle. So you localize it, you inject, and then it dissolves. Just to show you some pictures, this is the idea. No matter what you do, this is like the leopard tortoise. It'll turn over like this, and then it shoots the drug out. And the idea, again, depending how much sugar you put in, we can make the spring, we can control the spring actuation. This is what happens with the spring. I mean, once it's released, it just shoots the needle out just like that. And releases it in less than a millisecond. And the perforation risk, because you might be worried about, that's mitigated by controlling the actuation force. In fact, Gio points out that in gastroenterology, they already do things with what's called a Carlock needle. This doesn't do anything worse than that. This is like 4N force, and here's 9N force. But if you look at histology, there's really no damage. And this just shows that the insulin's stable. The ones that the team designed, more than 4 times longer than a liquid formulation. We've done it actually with a variety of different proteins and other molecules. These are just 2 of them. They've also done it with antibodies, but they're quite stable. And this is insulin now in pigs in this paper we wrote in Science earlier this year. And the point is that you get similar plasma levels when you give it compared to, in other words, if you give the pig an injection in the skin, the same system as you do in the gut, you get exactly the same amount. And if you look at efficacy, again, this is blood glucose changes. Again, if you compare intragastric, subcutaneously, and what we call a soma device, again, you get the same results. And again, this is showing no tissue damage. And it's worked reasonably well. We're still early, obviously. It has not been tested in humans yet, but it's an example of what might come in the future. So another thing, too, that I learned when I was at the hospital, one of the things I was curious about, yes, we do a lot with materials, and one of the things that I was curious about when I was working in the hospital was how do materials find their way into medicine? I thought very naively that it must be chemists or chemical engineers or material scientists that do that. But what I found is that was rarely true. Almost always, when I looked into this, the driving force for materials to find their way into medicine were medical doctors. And they urgently want to solve a medical problem. And the way they would do it is they asked, well, what object in my house kind of resembles the organ or tissue you want to fix? I'll give you a few examples. So in the case of the artificial heart, in 1967, some of the clinicians at NIH, they want to make an artificial heart. And they said, well, what object kind of like a heart, has a good flex life? And they said a lady's girdle. So they took the material in the lady's girdle, that's a polyether urethane, and that's what they made the artificial heart of. That was 1967. The only thing is now we're 52 years later, that's still what it's made out of. And if you start to think about why hasn't it worked very well, I mean, you can even read the Boston Globe, and why it hasn't worked well is sometimes blood is the surface of the artificial heart, the lady's girdle material. It forms a clot, and that clot goes to the patient's brain. They get a stroke, and they die. But if you think about it, something was designed to be a lady's girdle, you know, it may not be the optimal blood contacting material. And this problem, as I would see it, really pervades almost all of medicine. Almost everything was taken from your house, like dialysis tubing, vascular grafts, breast implants. Actually, one of those was even a mattress stuffing. So one of the things that you learn in chemical engineering is design. And so what we started doing, I started thinking about this back around 1980, is rather than just take something from your house, what if you ask the question, what do you really want in a biomaterial from a chemistry standpoint, biology standpoint, and engineering standpoint, and then could you design it from first principles? So I'll just pick one example, but we do a lot of this in the lab for different things. So when we started, the only polymer that were FDA approved were polyesters. And they dissolve like this. Starts out like this, and that were degradable. They start out like this, they get spongy, and then they fall apart. That's okay for some drugs, but you might imagine if you had like a toxic drug like insulin or an anti-cancer drug, it could dump out, and that would be very unsafe. So we said, from an engineering standpoint, how do you want the polymer to dissolve? So we said, rather than dissolve like this, what we want to build into it is dissolve from the surface first. Surface erosion like this, layer by layer erosion. How would you do that? So I apologize to the non-chemists in the audience, but I'll just go through a little bit. So we started to ask different design questions. First question is, what do we want? I'll just show a picture of a polymer schematic. And just, again, when you hear about polyethylene, that means all these monomers are ethylene. But if you heard of a polyester, that means all these bonds are ester bonds. So we started saying, well, if we want to really get this kind of surface erosion, what do we want the bonds to be? I'm sorry, the monomers to be. So we said, well, we could think about this different ways. First, what should cause the degradation? Should it be water? Should it be an enzyme? Should it be something else? And we started thinking about if we want it to be reproducible, all of you may have different enzyme levels, and your enzyme levels may change over time as your cellular response to a material changes over time. But all of you have excess water. So we said, let's build into this water to cause the degradation. But then we'd say, if we do that and we want to keep the degradation to the surface, what we want is these monomers, we said, to be very hydrophobic to make sure water can't get in easily. And then we go to the next question. If you do that, how would we still make the polymer dissolve fast enough? So now we go to the bonds, and we said, well, we want them to be pretty labile. And so if you go to organic chemistry, you could go from very stable to less stable, and we estimated we'd want to go up here to the anhydride bond. And therefore proposed the idea of hydrophobic polyanhydrides. And then what I did is we got people in the lab together, like Howie Rosen and Bob Linhart on the one hand, and then toxicologists and chemists like Mike Marletta, who was I mentioned before, on the other, to really write on the blackboard what kind of structure might meet these different requirements and still be safe. And so we picked this type of polymer. This is a copolymer. It would have an anhydride bond here. This is very hydrophobic. This is also very hydrophobic, but a little less so. So this is a structure we wrote on the board, and we actually then synthesized the polymer, how we did that, and basically by simply changing the ratio of those two units from, say, 0 to 79%, not only did we come very close to getting this kind of surface erosion, but we could really make these polymers last for anywhere from maybe 2 weeks to literally many years by simply tuning the ratio of those polymers. And these systems are really thin, even then, like maybe a millimeter thick. Well, if you could do that, then again, one of our goals really is to not just do the science and publish it, which we do, but also to try to hopefully do some human good. And in 1985, Henry Brem, who was just starting at Johns Hopkins, came to see me, and he wanted, one of the major things he was doing was treating patients who had brain cancer, that's glioblastoma multiforme. And these were some statistics he showed me that basically no matter what you do, the mean lifespan is less than a year. At that time, the only drug that people used to treat brain cancer was this one. And what Henry asked me is, could we come up with a new kind of therapy? Basically what we'd call local chemotherapy. Could you, rather than give the drug all out throughout the body, put the drug in a polymer wafer and then could he, as a neurosurgeon, he's going to operate in the patient anyhow, take as much of the tumor as he can out anyhow, but then before he closes the patient up, he would like to line the surgical cavity with little wafers containing the polymer and drug. Now, normally the drug lasts 12 minutes, but the polymer protects it. And Henry said, well, could we make it last from things he'd done for about a month? So we did that by simply adjusting the copolymer ratios. But then what happens is when you give this kind of therapy, you get high concentrations in the brain where you want it to be, but low concentrations in the rest of the body where it might cause harm. Well, one of the things you have to do if you're in academics, as everybody here knows, is you have to get money. You have to get money to solve these problems. So we would write grants to like NIH and other places, a lot on polymer chemistry and other things, and we did just terrible. I actually saved a number of the reviews. So when we first proposed this, the chemist said, well, these polymers can't be synthesized. But I had a very good graduate student then, who I mentioned before, Howie Rosen. Howie would later become president of Alza Corporation, which became a $12 billion company, and it's been elected to the National Academy of Engineering, and he was able to synthesize them. So we sent the grant back, and the reviewer said, well, that's good, but the polymers will react with whatever drug you put in. But I had another postdoc, Cam Leung. Cam's now as a chaired professor of bioengineering at Columbia University, also been elected to National Academy of Engineering, and he showed that there was no reaction. So we sent it back again, and the reviewer said, well, that's nice, but the polymers are what we say low molecular weight. They're fragile, so if they're going to break in the body, we still shouldn't fund it. But I had another couple postdocs, Avi Dome, who later became, actually now he's Dean of Pharmacy at Hebrew University, and Edith Mathiowicz, who's a full professor at Brown University, been elected to the National Academy of Inventors, and they made polymers that were higher molecular weight and very strong. Sent it back again, and the reviewer said, well, okay, that's good, but we still shouldn't fund it because nobody puts new polymers. They're not going to be safe at all. But Cato Lorenzen, another one of my graduate students, he'd later become Dean of Medicine at the University of Connecticut. Actually, he got the National Medal of Technology from President Obama a few years ago, been elected to both the National Academy of Engineering and National Academy of Medicine, showed that it was very safe. So we sent it back again. I'll stop pretty soon. We sent it back again, and the reviewer said, well, okay, but the drug won't diffuse far enough to kill the tumor. But I had another graduate student, Mark Saltzman. Mark actually became the inaugural head of the bioengineering department at Yale University, also been elected to the National Academy of Engineering and National Academy of Medicine, and he showed that it did diffuse far enough. Anyhow, this kept going on until 1996 when the FDA approved it. It was actually the first time that the FDA approved a new treatment for brain cancer in over 20 years, and really the first time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm speaking that I'm very, very proud of how well all the MIT graduate students and postdocs did. All of them have received enormous honors, have become deans, head of universities, elected to all kinds of national academies, whereas reviewers at the other schools, they didn't do that well. I'd actually like to show you what this operation looks like, but again, if anybody's squeamish and doesn't like the sight of blood, don't look. But just quickly, here's a wafer going into the brain. Usually they put 6 or 7 in and close up the brain. You know, it's very hard for me to get good advice when I give a talk, but a number of years ago I was giving a lecture at MIT, and my wife, Laura, who's here today, you know, I said to her at the end of the talk, because she came to it, I said, well, Laura, what did you think of the talk? So she said, well, Bob, you know, the talk was all right. That, by the way, is incredibly high praise. But she said, you know, I don't know if you realize when you had those 2 slides on, you were talking to all these chemical engineers, and you left those 2 slides on for over 10 minutes explaining every detail of the operation. And she said, I don't know if you were looking, but all those chemical engineers were turning green. So I always do what she says. So ever after that, you know, whenever I've given a talk, I give a warning like I did, and don't leave them on for very long. But I did want to tell you just the sequel to that story, which was about 2 years later, I got asked to give this dinner speech. And it was to a group of neurosurgeons. Actually, it was at the aquarium. So I gave the dinner speech, and I did the same thing. I gave the warning and everything else. And then at the end of the dinner speech, a number of neurosurgeons came up to me and they said, you know those 2 bloody slides you showed after dinner? I said, yes. They said, those were fine. They said, no problem. You could have left those on as long as you want. They said, but those chemical formulas, you know, to leave those on for a neurosurgeon is terrible. So anyhow, just to show you a little bit about the clinical data, there's certainly no cure, but this is from a neurosurgery paper. This was one of the early clinical trials showing that you do get about a 5-fold increase in survival after 2 years. This was actually much more recently, which is a summary of actually 60 publications, 60 clinical trials, showing again that you get significant survival improvement. Again, no cure, but using this kind of approach. And maybe most importantly, what this began to do is create a new paradigm where you could think about local delivery. And so actually, it's now being used in different kind of cancers, but probably where it's had the greatest impact, and this is how you can never predict how science works, actually turns out not to be in cancer as much at all, but tremendously in what's called interventional cardiology. In fact, some of this has been, what I'm going to tell you was done by Elazar Edelman. So people who was another one of my students and now head of IAMS at MIT. But what happened is that people may know if somebody has heart disease, one of the most common treatments is to give them what's called a stent. It's like a Chinese finger puzzle to keep a blood vessel open. The problem is when you do that, about 50 or 60% of the time you get a problem called restenosis. The blood vessels close because of smooth muscle cell proliferation. So what people now do to stop that is they put another anti-cancer drug, in this case Taxol, in a polymer coating, and that slowly releases it for, say, a month. It's basically the same idea, but it stops the smooth muscle cells from proliferating, and these are used in millions of patients every year. In fact, if anybody has heart disease, this is probably one of the most common treatments. I thought I'd end on just a couple minutes of one other thing we've done, which was mentioned, and that is to try to create new tissues and organs. And here I had the good fortune when I was in Dr. Folkman's lab of meeting Jay Vacanti. And after Jay worked with us on the angiogenesis problem, he became head of the liver transplant program at Boston Children's Hospital. This is one of his patients. And I remember Jay coming to see me, it must have been around 1982, 1983, after he had patients like that, and he said, well, is there some way we can do better rather than just do a transplant? And can we make tissues and organs from scratch? And the idea we came up with it was that maybe we could do this. This is from a paper we wrote in Science. It's been cited over 7,000 times. But basically, the idea is that you could take any, we wrote this before people talked about stem cells, but the idea is you could theoretically talk about almost any cell type, and if you inject it into the body at random, not much happens. But cells are smart. People like at Berkeley, at Basel and others, they've shown that cells, if they're put close enough together, can actually reorganize and create tissue structures. So you could take, for example, mammary epithelial cells, and if you put them close enough together, even in vitro, they can make what are called acini and make milk. So we wanted to give them a template, and so we would have a polymer system to create that template, and we'd have a bioreactor where we'd grow those, and the right forces end up being important in really making these tissues. And then theoretically, we could make virtually any tissue or organ we want. And just to show you some pictures, we've synthesized a number of different polymers to do this, but this is one of the polymer scaffolds with liver cells. And what you could do, this was done by another student, a postdoc of mine, Prasad Shastri. You could actually take ways of doing CAD-CAM techniques to make virtually any tissue or organ you want. That's computer-aided design. And here's an example. Let's say you want to, and this is pure speculation on my part, but let's say 40 years from now, somebody comes to a plastic surgeon and they want a new nose. So my speculation is that they'll have a computer screen and you could have about 50 different noses you could choose from. And to that end, Prasad designed like a nose. This is a polymer scaffold, 98% porous, and you could take the patient's own cells and make the nose. Now this is like a regular nose, but you could easily imagine if somebody wanted an upturned nose, you'd just take a little bit of this off. If somebody wanted a hook nose, I mean, they probably wouldn't, but if they did, we'd give them a little bit more, and then you could make the tissue. So I'll just show you a couple of examples of this. Here is an example with nude mice, which Jay and his brother were involved in, and later Gordana Vanyak and Lisa Fried, who work in our lab. Gordana's now a professor at Columbia, and Lisa is on the staff at MIT. And here this is a nude mouse, so it wouldn't get rejected. They redid this guy's skull, this guy's cheek, and if you open the animals up, it's pure white glistening cartilage. So it turns out, though, if you do histology, it actually looks like really good cartilage. And it isn't bad, but it's not, if somebody's a runner and they think we can help them, we're not there yet. But you can still do a lot of cosmetic things. So the Army actually approached Jay and Linda Griffith, who was one of my postdocs, now also in the faculty of MIT, and asked us, could we make new ears for people, soldiers coming back from Iraq and Afghanistan? So basically, Linda made this ear. Here's the scaffold. If you look at a high-powered scanning electron micrograph, here you see it. It's got cells. Over time, this will totally dissolve, and you'll get all matrix and cells. And this has not yet been put on patients for the ear, but it has been put on rabbits. Here's a rabbit with the human ear. And the reason that it was done on animals was to show it was safe. And here is actually the first patient that was treated this way. This was one of Jay's patients. And and if you look at him, he's a 12-year-old boy at this time. Now he's over 20, but he has no chest covering his heart. And like other 12-year-olds, he likes to play baseball. But you could imagine if he ever got hit with a baseball in the chest, he could die. So Jay operated on him, took a scaffold, and basically made him a new chest, and he's doing fine. Technology's also been licensed to different companies. One of them, several actually, have made new skins. I'll just show you an example of that. Here's a 2-year-old boy who's very badly burned. You can take the product, which is a neonatal skin fibroblast in this case on a polymer scaffold. You can put it in the child at the time of injury. But let's come back 3 weeks later. He's doing better. And 6 months later, he's pretty much healed. So you can use this for and our lab, in conjunction with others, has been working on many other tissues and organs as well, including spinal cord repair, hearing loss, vocal cord repair, new intestines, and so forth. The other thing that this would lead to is, or new blood vessels also. The other thing this would lead to is this whole idea of organs and tissues on a chip. And I thought I'd just end with an example of that was done by Milika Retzik, who's now, when she was a graduate student with us, and now professor at Toronto. And when she, so what we did, this is going to be hard on a chip. I'm sorry, let me go back. I'm not doing very well. Can we move that back? Oh, here we go. Yeah, so basically, if you put the cardiomyocytes on polymer scaffold and you look at it, you get something that has a very specific ultrastructure like a heart. And then you could put the cells on the polymer scaffold, and it literally beats 6 million cells beating at once. And I'll just show you the video on this. If you look closely, and it's not like we're shaking it or anything. It just beats. And what you can begin to do with these is actually Milika has a company on this where they can actually test this for new drugs for heart disease, both for safety and efficacy. And there are many, many tissues on a chip now being done all over the world, which hopefully someday will accelerate testing for different things, safety, efficacy, and so forth. Geo actually has an intestine on a chip, and other people are developing brains on a chip. There's almost everything on a chip right now. This just shows that you could take an EKG of this, even in vitro. So these are some of the things we've done. I mean, there are many others. I feel very fortunate that we've got like an amazing lab with just fantastic graduate students and postdocs and visiting scientists. To me, that's one of the most wonderful things about MIT. It's a super interdisciplinary lab, and you know, I just feel incredibly fortunate to be here and to be in many different parts of MIT, like the Chemical Engineering Department, the Koch Institute, Bioengineering Department, and Mechanical Engineering Department. And it's, you know, and really it's the students and postdocs. I could have mentioned a lot more names, you know, who are really responsible for this. I've also had a wonderful staff with Connie and Ilda and others. And most importantly, I want to acknowledge my wife, Laura, who's been kind enough to let me do all this. Thank you so very much for having me.
Thank you so much, Bob, for that inspiring talk and for being such a wonderful colleague over all of these years. I'd like to thank you again and also thank the Dreyfus Foundation for their leadership in recognizing this extraordinary member of our community and for all that the Dreyfus Foundation does to support scholarship in the chemical sciences. Now, for members of the faculty and Dreyfus guests who were invited to our reception, it will be held in the Koch Institute's lobby. And if you're not familiar with our campus, there are MIT staff waiting outside who can help direct you there. I'd like to thank all of you for making time to be here and hope you have a wonderful evening. Thank you.